everything that has a beginning has an end. This is the last lecture. So let's continue our chaotic journey with the next slide. So we were wondering what could be the recipe for an intelligent life. Okay. Okay. Who are we? We are just me and my friends. Okay. So we started thinking, what do you need to make a simple model of evolvable brain? Why this problem is interesting? Because it connects everything that I have talked about, and you will see how it connects things. Okay, I will show you some stuff. So you need nonlinearity because you expect brain to be rich, to have rich dynamics, and you get rich dynamics through nonlinearity, right? We expect it to be critical because the experiments are showing it, right? And also, when you are at criticality, you are between order and chaos, which means that you can form orderly ideas, but also you can be you can have rich ideas because of the chaos. It has to be plastic, meaning that it has to be adaptable to environment and be able to learn. So the connections need to be adjusted, etc. This kinds of stuff. So I'm going to talk about a master's thesis done in my group by Abdurrazak Efe. So as I mentioned, the goal was simple evolvable model of a brain. We had a lot of thoughts, but all those thoughts converged on one person's previous work. Okay. And that's Eugene Izhikevich. So here are his papers. Let's look at, before we go there, we already seen Fitzhugh Nagumo, right? Let me also talk about Hodgkin-Huxley model. Hodgkin-Huxley model, conductance-based model, is a mathematical model that describes how action potentials in neurons are initiated and propagated. Are you following? It is a set of nonlinear differential equations that approximate the electrical engineering characteristics of excitable cells, such as neurons and muscle cells. It's a continuous time dynamical system. And Hodgkin and Huxley did it in 1952 and got Nobel Prize after 11 years for this work. And they did it by looking at giant, let's also go there, squid giant axon. It's an axon in a squid, the marine animal. And it's giant, it's very large, 1.5 millimeter in diameter, typically 0.5. Okay, so you can play with it because it's big. So they did some experiments on it, then they realized that they can describe what's happening underneath by this model. The model is this, you see? It's like current that you apply, and it's a coupled differential equation, and you can see that it's nonlinear. There is n to the 4 m cubed, and also these VMs, etc. They are nonlinear stuff. Okay, very good, very good. But then comes Izhikevich, and he comes up with this simple model of spiking neurons. Okay, he describes that bifurcation methodologies. I will show you this book. This is a very nice book. enable us to reduce many biophysically accurate Hodgkin-Huxley type neural models to a two-dimensional system of ordinary differential equations. Remember, in 2D, you just have a limit cycle. That's the best that you can get, right? No matter how nonlinear you are. Nevertheless, this model is not just 2D differential equation. It has this mapping part as well. So there is an if statement which introduces huge nonlinearities. Remember, even if you have one-dimensional map, you could go to chaos, right, with logistic map. This is a 2D flow interrupted with a 1D map, right? Are you following? Do you see how it's at the essence of the course and it's connected to everything else? Very good. So basically, V is the voltage the membrane voltage that we see in neurons, and U is just control parameter. Look. So if this is the model, two coupled differential equations. If V is 30, then V is reset to C, and U becomes U plus D. Now, Hodgkin-Huxley model is very complicated. I think it has at least 16 parameters that I know of. Not that I know the parameters, I just know that there are at least 16 of them. This one has four parameters only, A, B, C, D, okay? Now with these four parameters, so this is parameter A, this is parameter B, this is parameter C, this is parameter D, you can actually produce nearly all kinds of biological neuron behavior. Like, do you want a regular spiking neuron? Then you should set A to 0.02, B to 0.2, C should be, where is regular spiking? C should be minus 65, D should be 8. I hope I'm making sense. Do you need a thalamocortical neuron? Not a problem. Just go and see where you should put TC. I mean, A is 0.02, B is 0.25, C is negative 65, and D is 0.05. So practically the only difference is this one is really low. Why this is really low? Well, there is some reason, and Izhikevich understands it very well because he has a beautiful book about it. Look. Just look at 500 pages of beauty. Just look at the cover. Do you like it? Then I presume this is Yelizaveta and Yekaterina, right? Liz and Kate. Anyways, let me show you the pictures. This is Izhikevich himself. They get fired. Yeah, I think the point here is that you put current in it. I developed the most realistic neural network. I do not see any neurons here, only equations. Neurons are dynamical systems. Are you happy with this? Look at these pictures. They will get much more nicer. Look at this. Look at this. This is amazing picture. So resting state potential is somewhere here because we know how potassium and chlorine ions are affecting this, and sodium ions. So during the spike, you take in a lot of sodium, etc., that kind of stuff. So you see how resting potential jumps to this value, etc. Are you excited? Blah, 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 blah. Hodgkin-Huxley explained in the best way possible. I want to show you this other parts. Look, practically what we have seen with this limit cycles, right? And we even have this one. Do you remember this one? We even have that one because it's all relevant. Just today I was looking at this book and I thought that instead of this course I should have just done just this book as a course. It will be a very nice course. Let me just show you one more type of pictures and then I'm done. You see these cylinders in 3D, they look really nice. Do you see them? Let me find the one that I like. Okay, these ones. Hopf bifurcation stuff. You see how neurons, like when they go like tick, 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 bursting, and what's happening when you are doing bursting. You understand it geometrically. Very nice, very nice. So this is the description of the model. Let me show you another paper by Izhikevich. Which model to use for cortical spiking neurons? A paper that, and the answer is my model. Why? There's a very good point. Look at this plot. Let me start from this plot. Biological plausibility versus implementation cost. Okay, Hodgkin-Huxley is quite biologically plausible, but prohibitively costly to implement, right? While Izhikevich model is here. Do you see? If you go even a little bit lower, you lose so much of biological plausibility. How does he calculate biological plausibility? Well, he has these biological things here, like tonic spiking, phasic spiking, phasic bursting, mixed mode, etc. Okay? The only thing missing with Izhikevich is biophysically meaningful. It's not biophysically meaningful. But otherwise, it gets all pluses. Do you see? Even Hodgkin-Huxley is missing these two pluses. Three pluses.
Everything right except it is not biologically meaningful.
Exactly. Exactly.
It's small triviality.
Yeah.
It's not by existing anyways.
Yeah, very good. Where am I? Okay, let me come back to here. So, as a single neuron model, we used Izhikevich model, of course. Now, let me talk about plasticity. So, plasticity, there are three kinds of plasticity: short-term, long-term, and evolutionary. Now, the short-term plasticity is due to Henry Markram. Have you heard of him? He was at the top of the $1 billion European Union project, Human Brain Project. Actually, my other master's student went there. I'm proud of him. Also, so with short-term plasticity, basically the way that I understand it is that neurons get tired when they are fired too much. Okay? Actually, not the neuron itself. You know, when a neuron fires, there's an action potential and it goes until it reaches the synapse, right? So there, everything happens chemically. Okay? The signal is transmitted chemically. So how chemically? There are like packages of neurotransmitters that come out of here and absorbed on the other side, right? But there are limited number of packages. So if you send too many firings, it will not be able to produce as many neurotransmitters. So your effective bond will go down if you use that bond too much. And this, in my humble opinion, produces also the criticality. If you are a bit super critical, like if you are firing too much, this being tiredness, it's called synaptic fatigue, will bring you down to critical point. Does it make sense?
Yeah.
Very good. How about long-term plasticity? This we also stole from Zhikevich. Let me explain what's happening here. So let's say this neuron is trying to fire, the neuron 1 is trying to fire the neuron 2. Okay? Let's say this is the spiking of neuron 1, and this is the spiking of the neuron. Let me just copy this. Now, if it's like this, this connection makes a lot of sense. I'm presuming that this is an excitatory connection. So 1 excites 2, okay? Now this makes sense because every time this gets fired, after a little bit of time, the other one gets fired. Do you see it?
Yeah.
So spike timing dependent plasticity increases this connection because this is a good timing. I fire, I want to excite you, and you fire. But if it's, oops, not too much. So this is a causal connection. 1 causes 2 to fire. This is a bad connection. I want to excite you. You get excited before I excite you. Yes? So my connection to you that is supposed to excite you is a pointless connection. So that connection goes away. This way, we basically create neuronal groups. If you have spike timing dependent plasticity, it automatically creates neuronal groups that fire together. Okay? It's like a thought. But it's an empty thought before you make it meaningful. It's just neuronal group. Let's say you learn something, and that neuronal group corresponds to what you have learned. Am I making sense?
Yeah.
No? In front, you.
I guess.
You guess. So it's like I'm teaching you now and you learn, but if you are learning before coming here, I haven't taught you, so I'm a pointless teacher. It's like that. No. Spike timing dependent plasticity works like that.
Okay.
Yes. Mm-hmm.
So in order for 1 to excite 2, 2 needs to be excited first somehow.
No, if we want 1 to excite 2, 2 should be excited after 1, so that this connection, this excitatory connection makes sense. Otherwise, it's meaningless, right?
But here 2 is...
Yeah. This is the wrong connection, so this connection will go away. But this connection will get improved, okay? However, don't forget that it's usually not 1 neuron firing the other one. It's usually hundreds of neurons firing others, right? So there could be synchronization problems. So you just let it be and you get your neuronal groups, okay? This is a way of learning, spike timing dependent plasticity. you can see that it's very easy to decide what to do with excitatory connections. However, there are also inhibitory connections. It's much harder to think about what to do with spike timing with inhibitory connection, because do you see the problem here? How do you measure inhibition? The measurement is that it shouldn't get excited. But then how do you know that it didn't fire because I inhibited it, or it didn't fire just because it usually doesn't fire? You see? The inhibitory part is, like, if you look at the literature, there is no consensus. There are review articles that just show 10 different kinds of spike timing dependent plasticity for inhibitory neurons. Okay? Very good. Then there is, okay, very good. So can we use, this is, by the way, yet another Izhikevich paper, solving, I didn't open the spike timing paper, but you can, I described it. In this paper, they are solving the distal reward problem through linkage of spike timing dependent plasticity with dopamine signaling. Are you still with me? Yes? So what is this? Here the problem is that we want, let's say you did something good, okay? How do you learn? How do you learn to do it again? Well, if you did something right, like I wanted water, I reached for it, I took it, here's my reward. But it came late. You know, I had to do this, take it up, bring, and then it's good, right? So there is a distal, what was the problem? Solving a distal reward problem. Are you with me? Yes? So what do you do? Here's what you do. How was it? Okay, I remember. Let's say you have another graph here. Another graph. It's called eligibility. Eligibility. Okay? So eligibility is low right now for this connection. And then it gets the correct way of spike timing, in a sense that first something fires, then the other one fires. So it's a good spike timing thing. So the eligibility increases suddenly, right? Why? It spikes, but then it starts decaying. Before it finishes, if you get the dopamine, then that connection increases, so that dopamine can reward previous actions. Good?
So this is for medium-term rewards.
Medium-term rewards. Very good. Very good. Are you still with me? Yes. So that's how you learn. What else do we have? Chaotic stuff, but very good. Then there is the morphogenesis problem. So our idea initially, we were really idealistic about this. We thought that we will have a code. It will start with one cell, and it will turn into a brain with its all connections, and then it will do very good things. So that was why we were interested in morphogenesis. I've shown you morphogenesis, so I'm skipping it. But then we realized that there is a neat way of doing it, and the algorithm is called NEAT, Neuroevolution of Augmented Topologies. Okay? So
I am 85% certain they found the acronym first.
Very good, very good. So basically, NEAT algorithm helps you do the following. You can still start with a single cell or a few cells, doesn't matter. But you can do the evolution in a sense that let me think about it. So there's a good organism A, it has some brain. Good organism B has some brain. You can augment those topologies together. Like you can take good, just as I explained in the genetic part, you take good things from this parent and put it on the other one. And this algorithm also allows the brain to grow. Okay? So we use that. And yeah, all this for solving a very simple problem. We have an agent and it has sensory neurons. So they are receiving, let's say, light they are living in 2 dimensions. So when you live in 3 dimensions, you see 2-dimensional way, right? When you live in 2 dimensions, you see 1 dimension. So there's just 1 dimension of sensory neurons, 2 eyes, and 2 types of things. There can be a red pill and a blue pill, just like in Matrix. And this agent has dopaminergic neurons as well. So some neurons, when they fire, they produce dopamine. Okay? So it will learn genetically what is pleasurable, what is not pleasurable. Okay? And there are interneurons, there are motor neurons that would make it move. So here's the first generation. Sorry about the Turkish titles. Ready? Now this is dopamine. This is success. So far we are already in -1. Now it will see these 2 things, red and blue, and it's just born in this world, and there are just 2 objects that it can see, and it doesn't know anything, so it doesn't do anything logical. It just hangs out. This is our organism. Just random motor neuron firings doing this. You see that it's not doing anything. Now, okay, it saw something, it moved suddenly, but it didn't go for it. Now, the other thing with this model is that in some worlds, red is the true pill. In the other worlds, blue is the true pill. It has to learn by eating and not feeling good. Does it make sense? So, let's say target is red. Now it's last generation, so it should go for red, but eventually, not at the beginning. At the beginning, it doesn't know. It's just born. It doesn't know what is good, what is bad. Okay? We just make it taste bad. So it goes for blue. You saw maybe some dopamine decrease there. Then it understands that it should go for red, and it starts avoiding blue, and it goes for, sometimes it just gets blue because, oh, now it's confused. On the average, I wouldn't say it's a perfect model, but that makes it beautiful.
Something weird.
What is it? Yeah, but it in the end. Okay. I will show you its success in the end. It's not perfect, but it was rising. Well, the thing is that before we perfected this model, Abdurrazak graduated. So
The weird thing I noticed was that it usually goes for the red, but sometimes the red spawns very close to it, and it gets red 5 or 6 times in a row in a rapid succession. But after that, it goes either for red or blue.
Yeah, maybe it gets too much dopamine.
Yeah, I mean, it's like it gets dopamine for free. It's almost like free dopamine there. I mean, the red spans almost at it, so it just random moments get rewarded almost. I wonder
Makes
if
sense.
it's something like instead of red and blue spawning randomly during training phase, When one gets eaten, then they both spawn at and some distance away from
yeah,
it.
that would make total sense. Should have thought about it. Yeah, I can pick this problem up again anytime soon, so I will consider that. Look, now it's the blue is the target, and although it tastes red several times, it finally decides on blue. Trust me. Did you see it avoiding red? Okay, not always. Blue, 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 red, blue, red, red, but blue. The point is that it goes up on the average. Okay, this is the result, like best fitness, average fitness, both of them go up high, but controls just stay down. So it works, right? If we let it evolve until today, maybe it would be started talking. But yeah. What is the point? The point is that whatever we saw so far is quite relevant for computational neuroscience as well. Okay? Do I have any other papers? Yes, I do, but I think we can stop here unless you have some questions.
PDF seems oddly fitting considering the deadline for the project is near it.
What?
Panic paper.
What paper?
There's a panic.pdf.
Ah, panic. Yeah, I didn't mention this. This is just human mobility behavior, like what happens when people panic. Like they get squeezed, and I think the solution was to put, so I as far as I remember, if you put some obstacle in front of the door, then it gets better. Like counterintuitive stuff happening. I would talk about this in previous lectures more, but I stopped talking about it. But the point is that, just like you said, don't we have applications in more social sciences kind of things? Yes, of course. Like, we didn't explore this map all the way. For example, you have farmer here, right? He started as a chaos scientist, but then he went on and built this company. This guy. Complex system scientist, entrepreneur, econophysics, that's the keyword here. Santa Fe Institute. Santa Fe Institute, where they came together, you know about it. And where is his prediction company? Prediction company was founded in Santa Fe. Doyne Farmer, this is another, he has cellular automata papers, this guy. The company used forecasting techniques to build black box trading in financial markets, statistical learning theory. Prediction company was shut down in 2018. In 26 years of operation, prediction company had only one losing year, 2007. Okay? Speaking of econophysics, if we go to Scholar and just ask for complex systems. This is not the way to ask, right? Should we ask? I forgot how to ask this. Anyways, I know the guy's name, so let me ask like that. Eugene Stanley. Do you see? phase transitions and critical phenomena. He has a book on this. Fractal concepts in surface code. Do you remember this article from somewhere else? It was in Barabashi's profile, the network guy. So this is his teacher, as far as I remember. And it has...
Textbook.
Hmm?
It is an economic physics textbook.
Yes, exactly. Introduction to Economic Physics, Correlation and Complexity in Finance. So, it's there, right? What else do we have here? I mean, I don't know these people because it's just a little bit out of my interest. But any other questions? So it has huge applications in social sciences as well, and Santa Fe Institute is still there. If you want, you can go there. It's in the middle of the desert.
By the way, I have a question about that pen paper, that stampede thing.
Say it again.
That stampede paper about panic.
Ah, yes.
Can these ideas be used to, fasten up the traffic as well?
Yeah, absolutely.
So is it possible that a road like that, where there's a curve for
Yeah.
be faster?
Yes. I mean, if you go there, you will find plenty of articles on this. Like, it doesn't just stop with human walking, it's also traffic. They also did a lot of stuff on traffic. You are interested in traffic, right?
Yeah.
Yes. Did we talk about Rule 184?
184, I don't think we did in context of traffic.
Rule 184. Remember, it was cellular automata thing, and can be used in a simple model for traffic flow. This could be a good idea, starting point. Okay. Any other questions? No questions? Any comments?
About that map of complexity, were there anything about turbulence and fluid dynamics?
Yes. Where is Kolmogorov? Here he is.
In context of information there.
Yes, but he's there. Once Kolmogorov is there, turbulence is there. But yeah, more on the turbulence can be found in dynamical systems part, somewhere here, upstairs. I don't know if we have someone studying turbulence in particular, but the idea of self-reference, the idea of renormalization, they are all inside the turbulence phenomena.
Yeah. I mean, it's $1 million question for a reason.
Yeah. Any other questions? Let's stop here. Thank you very much. Have fun. Don't be late with the project submissions. Goodbye.